very familiar to you. You have already covered, okay, most of the content of this chapter during SPM. Okay, so here, as you can see, these are the learning objectives of this chapter, which is first you have to state the cell theory. Okay, so basically there are two cell theory that you have to know, and then it is frequently asked. Okay, the theory in the test, right? And then second, compare and contrast the structure of prokaryotic and eukaryotic cells. So you have to know examples of prokaryotic cells, example of eukaryotic cells. Okay, what are the differences between these two cells? Okay, and then for eukaryotic cells, it is divided further into unicellular and multicellular. And for multicellular eukaryotic cells, you have, for example, animal cell and plant cell. So for that, you have to know the difference between the structure of animal and plant cell. And then you have to know how to draw animal and plant cells, okay? And then the next learning objective is you to be able to describe and tell, okay, the function of organelles within the cells. Okay, so you have to know how to draw nucleus. You have to know how what is the function of the nucleus. You have to know what is the component of the endomembrane system. It has a 6 component. One of it is the nucleus. Okay, and then you have the rough ER, the smooth ER, the Golgi apparatus, the lysosome, the plasma membrane, and also the vacuole. So those are the component or the organelles of the endomembrane system. And then you have to also know how to draw the structure of mitochondria, chloroplast. You have to know the function of each of the organelles. You have to know what is peroxisome. Okay, so those are the organelles that we are going to look at for this chapter. And then also you have to know there are structure within cells. Okay, we call it as the cytoskeletons. Okay, cytoskeletons, which function basically to maintain the structure of cells. One of its function is to maintain the structure of cells. Okay, so there are 3 types of cytoskeleton. You have macrotubule, microfilaments, and also intermediate filaments. Okay, so those are the 3 cytoskeleton. Okay, so basically cytoskeleton are made up of proteins. Okay. And then after that, okay, we are going to look at specialized animal tissues. Okay, you have to know the different types of cells. Okay, that makes different types of tissue within animal, and same goes to plant. So you have to know the different types of tissue in plant and what are the function of those tissues. Okay. Can everyone clear my voice clearly? Dia dengar suara saya dengan jelas?
Yeah.
Jelas.
Okay. So for introduction, so as you know, all organisms are made up of cells. Okay, so if it's unicellular cells, it is composed only of one cells lah. Okay, such as bacterial cells. Okay, bacteria, or unicellular eukaryotes such as protists. So example of protists, you have, for example, amoeba. So amoeba is consists of only one cells. Okay, so it is unicellular organism. And then you have also other complex organism that is composed of many cells. Okay, so those are eukaryotes. For example, as human. Okay, human, we are composed of many cells. All right. And then the basic structural and functional units of every organism. So cells are basically the basic functional and structural units of every organism. So every cells will have their own function. Okay. So that they can carry out specific work or function. Okay, and then these are the basic features of all cells. Okay, so regardless it is bacterial cells or animal cells or plant cells, so all these cells, they must have plasma membrane. Okay, so what is the function of plasma membrane? It functions as a selective barrier. Okay, so it is to separate between the component within cell. Okay, within cell, and also to separate the component of within cells, which is the organelles, okay, within the cells, from the outside environment, which is the extracellular fluid. Okay, so it is a selective barrier. Why is it selective? It only allows certain substance to enter into cells. Okay, so, through either active transport or passive transport. All right. So that we are going the selective permeability of plasma membrane we are going to look at in chapter 3. Okay, and then the second basic feature of our cell is that all cell must have a cytoplasm or cytosol, which is a semi-fluid substance in which organelles and other components are found. Okay, and then after that, all cells must have chromosome lah. Okay, chromosome which function to carry genetic information. Okay, in the form of genes in the DNA. Okay, and then the last one, all cells. Okay, regardless it is bacterial cells or plant cells or animal cells, they must have a ribosome. Okay, so the function of ribosome is to make protein. So what is the function of protein within these cells? One of the function or the protein can function as enzyme. Okay, so, as enzyme, it is to catalyze reactions. Okay, that occurs within cells. Okay. So, and these proteins are made, okay, from the instruction from the gene within the nucleus. All right. So why is it that the basic feature of all cells does not include nucleus? Okay, so it is because for prokaryotic cells, bacterial cells, they do not have nucleus. Okay, so nanti kita tengok kenapa. Alright. So two concepts of cell theory. Okay, so this is the cell theory. So two basic concepts of cell theory is one, cells are the basic. Okay, cells are the basic living units of organization and function of all organism. Okay, so and then the second one, all cells come from another cells. Okay, so in order for a cell to exist, so it is made from pre-existing cells. Okay, through cell division.
Madam nak tanya,
Ya.
apa beza plasma membrane dengan cytoplasm?
Plasma membrane.
Plasma membrane dengan cytoplasm.
Okay. So this is a cell. Okay, so outside you have the extracellular fluid. Okay, fluid outside of the cell. And then this is the plasma membrane. Okay, the selective barrier. Okay, the selective barrier that separates, okay, the component within cell from the outside. Okay, so and then inside you have the nucleus, and the fluid within the cell here you have the cytoplasm or the cytosol. Boleh?
Boleh, madam.
Okay. Okay, and then okay, so cells or organisms are divided into two. One is prokaryotic cells, second is eukaryotic cells or prokaryotes. Kita guna istilah ada YOTS ni. Okay, prokaryotes or eukaryotes is referring to the organism. Okay, if prokaryotic cell is referring to the cell of the prokaryotes or the eukaryotes. Okay. Ni. So prokaryotic cell, okay, organism dia adalah prokaryot. Eukaryotic cell dia eukaryotic cell. Organism dia adalah eukaryotes. Okay. So example of eukaryotes is unicellular. It is only made up of one cell. So multicellular, many cells. Okay. So example of multicellular, you have animal cells and also plant cell. For unicellular, you have, for example, protists. Okay, so protists, example, you have amoeba, okay, or euglena or paramecium. Okay, so those are example of unicellular organism. So jangan mistake proteus tu adalah prokaryote, salah. Okay, so proteus, amoeba, paramecium, euglena kan, so they are not prokaryote, they are unicellular eukaryotes. Okay, so they are microscopic, they are very small, but the size is much bigger than the prokaryotes. Okay, so prokaryotes are, for example, bacteria. Okay, so so we are going to look at for each one. So prokaryotes, so what does it mean? Pro, it means before, karyote, it means nucleus. So these are organism that exist, okay, before organism that has nucleus. Okay, so they are more primitive organism. Okay, so they exist earlier than eukaryotes, more primitive, right? So organisms for prokaryotes, they are in the domain of bacteria and archaea. So consists of prokaryotic cells, which is a single cell microorganism. Okay, so the size are smaller and structurally are more simple than eukaryotic cell. Why is it much more simpler? Because they do not have membrane bound organelle. Do not have membrane bound organelle. Okay. So they don't have nucleus, they don't have mitochondria, they don't have chloroplast, they don't have lysosome. Okay, so they do not have any membrane-bound organelles. Okay, so that's why they are more primitive and much more simpler than eukaryotic cells. So examples, bacterial cells and cyanobacteria. So cyanobacteria are bacteria that is capable of carrying out photosynthesis. Okay, so these are the characteristics of prokaryotic cells. Okay, so they have no true nucleus. They do not have nucleus because why? Nucleus is a membrane-bound organelle. So the DNA of the bacteria is concentrated in a region, we call it as the nucleoid. Okay, the nucleoid. So this is the nucleoid. So this is the nucleoid where you will find the genetic material of the bacteria. Okay, so therefore the DNA of the bacteria is in direct contact with the cytoplasm. It is not separated by membrane of the nucleus. Okay, so why? Because they do not have any nucleus. So the DNA is confined within the nucleoid region. Okay, so no membrane bound organelles, as I mentioned. No mitochondria, no chloroplast. Okay, for example, so cytoplasm are bounded by plasma membrane and they are unicellular. So as you can see from the diagram, okay, so they have several wall. Okay, they have the plasma membrane, okay, to separate the inside of the cell from the outside, and then after this plasma membrane, you have the cell wall. Okay, the bacteria will have cell wall, and then it is surrounded by capsule. Okay, and also they have a membrane. What is the function of the membrane? As attachment structure. Okay, so to attach to some kind of structure. Okay, and then the second one is that as you can see from the diagram, the bacteria will have a ribosome. Okay, so the function of the ribosome is to synthesize protein. One of the function of the protein is that it can become enzyme. Okay, and then they have cell wall, and then they have capsule. Okay, so which is jelly-like outer coating of many prokaryotes, and then flagella, which allows the bacteria to be able to swim. Okay, locomotion flagella. Okay, to move. Okay, it allows the bacteria to be able to move. And then the next one, okay, these are the explanation for eukaryotes. So it means true. Karyotes mean nucleus. So it means that for this organism, they have nucleus. Okay, so the nucleus function to store the genetic information. It contains the DNA. Okay, so these are the characteristics of eukaryotic cells. So they are larger and much more complex. Okay, so why complex? They have membrane-bound organelles. They have many functions. Okay, compared to the prokaryotic cells. Okay. So they are much generally much larger than the prokaryotic cells. So this is the size of a eukaryotic cells, ranging from 10 to 100 micrometer, and then the DNA is within the nucleus. Okay. Sorry, saya batuk. Okay, so the nucleus, the nucleus, as you know, it has membrane. So the membrane of the nucleus, we call it as the nuclear envelope. Okay, so the nuclear envelope, it is a double membrane. Okay, it is a double membrane. Mana pen saya hilang. Okay, it is a double membrane. Okay, consists of inner and outer membrane. So they have eukaryotic cells have many other organelles that has membrane. Mitochondria, chloroplast, lysosome, okay, Golgi apparatus, those are organelles that have membrane. And then cytoplasm is in the region between the plasma membrane and the nucleus, okay, the fluid within the nucleus. And then eukaryote can be unicellular or multicellular. Okay, so these are examples of multicellular eukaryotes: plant cell, animal cell, and also plant and animal cells. Okay, plant and animal cells are multicellular eukaryotes. Okay, so as for unicellular, you have here, okay, unicellular eukaryotes. You have the protists, okay, protists, and it has in the diagram here you have the chlamydomonas. Okay, it is unicellular, single cell, and then they are microscopic. Okay, so in this table, okay, it gives you the differences between the prokaryotic cells and eukaryotic cells, eukaryotes and prokaryotes. So for prokaryotes, okay, the size of the ribosome, they are smaller, but as for eukaryotes, the size of the ribosome are much bigger. And then for prokaryotes, they have free ribosomes. Okay, it means that the ribosomes are scattered in the cytoplasm. Okay, they are not bound to any structure within the cell of the prokaryote. But as for eukaryotic cells, okay, so they have two types of ribosome, which are free ribosome. Okay, so they are scattered in the cytoplasm, and also bound ribosome, which is bound to the ER or bound to the membrane of nucleus. Okay, nucleus, bound ribosome. Okay. And then for prokaryotic cell, the cell division is generally by binary fission. Okay, binary fission is a type of asexual reproduction due to for prokaryote they do not have nuclei. Okay, but as for eukaryotes, okay, cell division involves mitosis, which involves, for mitosis it involves somatic cells. Okay, and as for eukaryotes, and the other one is meiosis, okay, which involve gamete cells. Okay, so istilah mitosis and meiosis ni adalah untuk cell division yang ada nukleus sahaja, pembahagian nukleus. So that is cell division for eukaryotes. Tapi kalau untuk prokaryotes, sebab dia tak ada nukleus, so dia akan undergo binary fission. Okay, ingatlah istilah mitosis dengan meiosis is for cell that has nucleus. Mitosis is somatic cells, all your body cells, okay, that except for gamete cell. So gamete cell undergo meiosis, right? And then prokaryote, the cell wall are made up of peptidoglycan, all right? And then for eukaryotes, plant and cell algae cell wall are made up of cellulose. The cell wall of fungi is made up of chitin, okay? And then for prokaryotes, so obviously the cell will have to generate energy, okay, through cellular respiration. But how to generate energy when they don't have any mitochondria? So the process of cellular respiration, it occurs in these structures, mesosome and also in the plasma membrane. Okay, so the plasma membrane of bacteria, for example, they have enzymes that are involved in cellular respiration to generate ATP. Mesosomes are folded, okay, folded membrane within the the plasma membrane of prokaryotes. Okay, folded membranes. Those are mesosome. Okay, and then for eukaryotes, no mesosome, but they have mitochondria for cellular respiration to occur to generate ATP, energy. Okay, energy to do cellular work. Okay. And then for prokaryote, so some prokaryote can carry out photosynthesis, such as the cyanobacteria. But how to carry out photosynthesis when they do not have chloroplast? So they have photosynthetic lamella. Okay. And then for eukaryote, photosynthesis is carried out by in organelles, which is the chloroplasts. Okay. And then two types of eukaryotes. You have unicellular eukaryotes and multicellular eukaryotes. Okay, so because they are for unicellular, because due to their small size, okay, so therefore they are microscopic. You can only observe this organism under the microscope. So example, you have the protists. Okay, protists which is protozoa. Amoeba, okay, paramecium, for example, some algae and fungi. So nucleus and other organelles surrounded by membrane. So unicellular eukaryotes, jangan ingat dia adalah bakteria. These are not bacteria. So these are unicellular eukaryotes. Sebab apa? Dalam sel dia ada chloroplast, membrane bound organelles, ada nucleus, membrane bound organelles, ada mitochondria, for example, membrane bound organelles. Okay, so they have membrane bound organelles. So this is the structure of a plant cell. Okay, so remember you have to know how to draw a plant cell. So this is a 3D structure of a plant cell. So bila dalam exam, kalau terror drawing boleh lah draw 3D kan, tapi tak perlu ambil banyak masa untuk ambil drawing. Just draw a simple diagram of the plant cell. Okay, so you have the cell wall. Okay, and then plant cell has a fixed shape. Okay, and then the plasma membrane, you have to label lah, cell wall, plasma membrane, and within this plant cell, you will have a large central vacuole. Okay, label lah central vacuole. Okay, plasma membrane, cell wall. Okay. And then because the plant cell have a large central vacuole, so the nucleus of the plant cell is located at the peripheral. Maksudnya due to the large size of the central vacuole, it pushes all the other organelles to the side. Okay, peripheral. The location of the nucleus here is at the peripheral, kat tepi-tepi cell. Okay, so the cytoplasm are pushed to the side. And so does the other organelles. Okay, so inside you have other organelles such as the apa yang ni? This is the mitochondria. Okay, mitochondria. Draw lah dia cantik sikit sebab saya draw kat macam ni kan. So mitochondria, they have folded in a membrane. And then you have what? Other organelles. You have the chloroplast. So chloroplast is also a double membrane organelle. So inside the chloroplast, you have thylakoids. Okay, thylakoids. So this is chloroplast. Okay, mitochondria. Apa lagi dia ada? So dia ada Golgi apparatus. Okay, draw lah the Golgi apparatus kan. Golgi apparatus, and then dia ada endomembrane system. Endomembrane system, the rough ER, the smooth ER. Okay, so draw it properly. Okay, so ada apa? Ribosome. Okay, these are free ribosome. Ada lysosome. Ada apa lagi ya? Okay, so those, but the basic structure is like this. Okay, so tak payah nak lukis dia 3D macam ni kan. Just draw a simple diagram. Okay, that is much neater than mine lah. Okay. And then for animal cell, so this is the structure of animal cell. As you can see, kalau tengok gambar ni dia macam kompleks dia nak draw, tapi sebenarnya tak susah pun kan. Senang je. Okay. So they have roughly a spherical shape. Okay. Because for animal cell, it does not have any central vacuole. So the nucleus is roughly at the center. So this is the nucleus. Okay. So nucleus is a double membrane organelle. And then you have the mitochondria, okay, and then the outer membrane of the nucleus is directly connected to the rough ER. Okay, rough ER is folded, and then from the rough ER you have the Golgi apparatus. Okay, the Golgi apparatus, and then you have the secretory vesicle, the transport vesicles, the lysosome, the ribosome. Okay, and then you have the apa nama? Ni. Centrosome, okay, centrosome, which is made up of centrioles, okay. So just draw a simple structure with label, right? So dalam exam dia suka tanya soalan-soalan yang suruh lukis structure lah, so you have to know how to draw, okay. Make sure you label. Okay, so this table, it shows you the difference between animal and plant cell. So for animal cell, they do not have any cell wall, but plant cell have cell wall. So the cell wall is made up of cellulose, right? And then obviously both cell will have plasma membrane lah. So dalam soalan, kalau dia tanya compare and contrast, okay? Compare and contrast, it means that you have to find similarities and differences between the two subject, okay? Plant or animal cell, for example, okay? So ingat compare and contrast, you have to find similarity and differences. So when the question asks you for differences, you have to do in a table form. Okay, so you have to compare apple to apple lah. Jangan apple to pear. Walaupun facts dia betul, dia jadi salah. Okay, so make sure you write properly. Okay. And then for plants, animal cell does not have any plasmodesmata. Plant cell have plasmodesmata. What is plasmodesmata? It is a junction, cell junction between cells. Okay, so for example, here you have the first plant cell and the second plant cell. Okay, so in order for the first plant cell to transport material to the second plant cell, it is through a connection between the two cells. So this connection here, we call it as plasmodesmata. Okay, so kalau tengok kat gambar ni, nampak ada lubang-lubang ni kan? Okay, lubang antara sel yang pertama ni dengan sel yang kedua ni. So you have connection, which is plasmodesmata. This is to allow the cell to transport materials from cell to cell. Okay, from cell to cell. So that is the function of the plasmodesmata. For animal cell, they do also have cell junction, but it is not called as plasmodesmata. They have other terms such as, for example, gap junction. Okay, so maybe boleh lah juga tulis. Instead of you writing there's no plasmodesmata and pigs, you can write for animal cell, they have gap junction. For plant cell, they have plasmodesmata. Okay, and then for animal cell, no chloroplast because they do not carry out photosynthesis. Plant cell, because they carry out photosynthesis, they have chloroplast. And for animal cell, they do have vacuole, but the vacuole, the size of the vacuole is much smaller. But for plant cell, they have central vacuole that is large. Okay, they have large central vacuole which is filled with cell sap. Okay, so this is to make sure that the central vacuole can store water, for example. Okay, so to make sure that the plant cell is always in the turgid condition. Okay, plant has to be for plant cell, the normal condition for plant cell is turgid. Okay, in order for the plant cell to be turgid, they have to store water in the central vacuole. Okay, so that's why the central vacuole is large. Okay. And then the membrane of the central vacuole, we call it as tonoplast. So for plant cell, because they have central vacuole, they have tonoplast, the membrane of the central vacuole. And because animal cell does not have any central vacuole, obviously no tonoplast. Okay. So the next one is animal cell. So the position or the location of nucleus within animal cell, they are located at the center of the cell. Roughly, okay, at the center, because there's no central vacuole. But as for plant cell, because they have large central vacuole, so all the organelles, okay, within the cytoplasm and the nucleus are pushed to the side. Okay, they are located at the peripheral side of the cell. Okay, due to the presence of the large central vacuole. And then within the animal cell, you have the glycogen granules to store glycogen. Okay, for plant cells, they have starch granules to store starch. And then animal cell, the size are smaller compared to, for animal cells, the size is smaller compared to plant cell. Plant cell have larger size compared to animal cell. And then for animal cell, they have cilia and flagella. Okay, so for example, in our body, okay, the cells that have cilia are the cell that line your trachea. Okay, as you breathe in, obviously the air that you breathe in is not clean, full of contaminant or pollutant. So the trachea will filter out, okay, the contaminant by having the cilia. Okay, cilia lining the trachea. Okay, to filter out any contaminant that tries to enter into your lung. Okay, and then the oviducts, okay, also has cilia. The function of the cilia lining the oviducts is to move, okay, move the secondary oocyte. Okay, secondary oocyte released from the ovary, okay, until it reaches the uterus. Kan? Ovary release apa? Ovum kan? Release ovum into the fallopian tube or the oviducts, and then the oviducts is lined with cilia. It is to move the ovum or the secondary oocyte until it reaches the what? The uterus. Okay, the uterus, right? And then sperm have cilia or flagella? Have flagella. Okay, flagella. So kalau dalam kalau sel, okay, satu sel ni, okay, kalau cilia dalam kuantiti yang banyak, kan, cilia. Tapi kalau flagella macam sperm cell, sorry tak cantik. Sperm cell, okay, dia ada only one flagella. Okay, cilia exist dalam kuantiti yang banyak untuk satu-satu sel tu, tapi flagella dalam kuantiti yang lebih sikit lah. Okay, and then plant cell does not have any cilia and flagella. Animal cell have lysosome. Plant cell, the function of the lysosome is to store hydrolytic enzyme. Okay, nanti kita tengok what is the function of the hydrolytic enzyme. And then for plant cell, lysosome usually absent except for insectivorous plants such as Nepenthes. Okay, Nepenthes apa tu? What is Nepenthes? Nepenthes ni adalah periuk kera kan. So dia makan insect. So it obviously kena digest insect tu. So they have lysosome. So lysosome has hydrolytic enzyme, okay, or digestive enzyme, okay, to digest the food. Okay, so next we are going to look at all the different organelles, okay, within cell. So this chart is to just give an overview of the different organelles that we are going to look at, which is the first organelle is the nucleus, and then you have the ribosome, and then you have the endomembrane system. Okay, so these are organelles that involve in manufacturing or breaking down organelles. All right, so nucleus. Okay, so from the nucleus it stores genetic information to manufacture what? To manufacture or to produce RNAs. Okay, RNAs. And then from the RNA is to produce protein. So manufacture something, okay, to produce something. Then breakdown, for example, lysosome contains hydrolytic enzyme. It is to break down food, for example. Okay, so the endomembrane system, it has 6 components. It consists of the nuclear envelope, the Golgi apparatus, the vesicles or the vacuoles, and then the endoplasmic reticulum. You have the smooth ER, the rough ER, and then the lysosome and also the plasma membrane. So these are the 6 components of the endomembrane system. Okay. And then you have the oxidative organelles, which you have the peroxisome. And then you have the next one, which is network of fibers structure, which is the cytoskeleton. You have the microtubules, microfilaments, and intermediate filaments. These are actually proteins. Okay, they are protein. They are structural proteins. Okay, structural protein to form structures within cell. All right. And then the next one, you have the extracellular structures, structures that are located outside of the plasma membrane. So you have the plasma membrane, and then outside you have the cell wall for plant, and then for animal cell, they have this one is for animal cell. Animal cells. This one is for plant cell. This one is for animal cell. Okay, just outside of the plasma membrane. And then you have the intercellular junction. Yang tadilah plasmodesmata, gap junction for animal cell. Okay, you have other types. Nanti kita tengok ada a few more. Desmosome, for example. And then the next one, you have the mitochondria and the chloroplast. They are energy converting organelles. Okay, so the chloroplast, because it carries out photosynthesis, convert what type of energy? From light energy into chemical energy, which is the ATP. Okay, so so that is energy converting organelles. Mitochondria from glucose, kan, chemical energy, chemical glucose, kan, and then to another form of chemical energy, which is ATP. Okay. Okay, so the first organelle here is the nucleus. All right. So for eukaryotic cell, it contains most of the DNA. Why is the statement says most? Okay, so because you can also find in the cell, okay, the DNA that are located in the chloroplast and also the mitochondria. Okay, so mitochondria and chloroplast will have their own DNA. Okay, and then the nucleus are usually spherical or oval in shape. And then in animal cells, okay, it is located in the central region of cell. Kalau punya nota tu, dia start dengan ayat ni kan? Located in the central region. The central region is referring to the animal cell. Sebab kalau plant cell, kat mana? Peripheral. Okay, peripheral. Because due to the presence of the large central vacuole. Okay. So for prokaryotic cell, they have no membrane-bound nucleus, but the DNA is located in the nucleoid region. Okay, DNA is located in the nucleoid region. Okay, so basically this is the structure of a nucleus. All right. So nucleus, as you can see, it has double the nuclear envelope. Okay, this is the nuclear envelope. Which is a double membrane. So the double membrane is the inner membrane and the outer membrane. The outer membrane, if you look carefully in the diagram, the outer membrane is continuous with the membrane of the rough ER. Okay, so the outer membrane of the nuclear envelope is continuous with the membrane of the rough ER. Okay, and the rough ER is continuous with the smooth ER. Okay, so that's why the endomembrane system, it includes the nucleus, the ER, the smooth ER, the rough ER, the Golgi apparatus, because the membrane are connected to one another. Okay, and then in the nuclear envelope, they have pore. Okay, pore. So the pore, we call it as pore complex. It is made up of proteins which are embedded in the membrane of the nucleus. Okay, and then you have at the center here is a dense part within the nucleus. We call it as the nucleolus. The nucleolus is the region where ribosomal RNA are produced, okay, and the subunits of ribosome are produced. That is the function of the nucleolus, the site where RNA are produced and the subunit of ribosome are produced in the nucleolus, okay, outside. And then the rest you have the chromatin, okay. So chromatin are composed of here DNA plus protein histone. Okay, so they will bind together to form the chromatin, and then from chromatin to chromosome. Okay, to chromosome. Okay, so each structure kita tengok satu-satu. So the nuclear envelope or the nuclear membrane encloses the nucleus, separating the the content of the nucleus from the cytoplasm. So it is the double membrane, inner and outer membrane, consists of phospholipid bilayer. Ingat ya, plasma membrane dan membrane organel yang lain adalah terdiri daripada phospholipid bilayer. So for nucleus, the nuclear membrane, they have pores. Okay, so this is to allow substances to enter and exit the nucleus. Okay, regulate entry and exit of macromolecules in and out of the nucleus. So it controls protein to enter into the nucleus, RNA to be able to exit the nucleus, for example. Okay, so it control or regulate the entry and exit of macromolecule between the nucleoplasm and the cytoplasm. Okay, nucleoplasm tu the fluid, okay, within the nucleus. Macam cytoplasm lah, cytoplasm dalam sel. Tapi kalau nucleoplasm dalam nucleus, okay. So the outer membrane is continuous with the membrane of the endoplasmic reticulum, okay. And then the nucleopore, okay, surrounded by protein structure called nuclear complex. Dalam opini nota tu dia cakap stud, okay, stud with ribosome. It means that you have, stud with pore complex. It means that you have protein embedded in the nuclear membrane to form the pore complex. Okay. So the inner and outer membranes are continuous at the lip of each pore. Okay. So nampak dia continuous. The inner and outer membrane, they are continuous. Inner and outer. Okay. So it is continuous. Okay, so the nucleolus, okay, plural nucleoli. So dalam nota awak it says not enclosed by a membrane. Statement tu tak berapa nak tepat lah. Sebab apa? Sebab nucleolus tu dalam nucleolus which is surrounded by nuclear membrane. Okay. Tapi maybe dia nak refer kepada nucleolus itself does not have its own membrane. Macam tu. Okay. So it is located within the nucleus. It contains DNA, RNA and proteins. Okay. So the nucleolus here, okay, it is where RNA are produced. Okay. And from the cytoplasm here, you have the cytoplasm. Okay, so in the cytoplasm you will have protein produced in the cytoplasm. So this protein will enter into the nucleus through the nuclear pore and enter into the nucleolus to form what? The large and small subunit of ribosome. Okay, the large and small unit of ribosome. Okay, so maksudnya protein daripada luar, daripada cytoplasm bawa masuk ke dalam nucleus, enter into the nucleolus where the protein will combine together with the rRNA to form the subunit of ribosome. So it forms the large subunit, the small subunit, and then these two subunits, okay, then exit the nucleus through the nucleopore, and then in the cytoplasm, okay, this subunit will assemble to form the ribosome, okay, to form the ribosome. Okay, so next is the nuclear lamina. So what are nuclear lamina? It is an array of protein filaments or intermediate filaments. They are actually fibrous protein, okay, that are the function of this nuclear lamina is to maintain, okay, the shape of the nucleus supaya nucleus tu tegak, maintain lah dalam structure spherical tu. Kalau tak ada nuclear lamina sebagai support, dia maybe crumble together kan. Alright. So the nuclear lamina is to maintain the shape of the nucleus. So it lines the inner surface of the nuclear envelope. Okay, so this is the filaments. Okay, it is structural protein to support the shape of the nucleus. And then inside the nucleus, you have the nuclear matrix. Okay, which is a network of protein fibers found throughout the nuclear interior. Okay, so both the nuclear lamina and matrix organize the genetic materials in the nucleus. Okay, it organizes the DNA within the nucleus, the chromosome within the nucleus. So next, you inside the nucleus, obviously you have the chromosome. Okay, so this is a chromosome. Okay, as you know, daripada dulu lagi bentuk dia macam ni lah, bentuk X, right? Okay, the chromosome, and then it contains the DNA. So this is DNA. Okay, so this is DNA. DNA is helical, and then because DNA is a large molecule, so your cell, your nucleus has to organize the DNA properly. So the DNA will coiled around a protein histone to form chromatin. Okay. So the single DNA molecule will coil around protein histone to form chromatin, and when the cell is about to undergo cell division, okay, so the chromatin condenses to form chromosome. Okay, so daripada DNA jadi chromatin sebab dia bind dengan protein histone, jadi chromosome. Okay, so chromosome, you can only see this chromosome when the cell is about to divide. So ini slide yang saya tambah sikit. So function of nucleus, one is to carry genetic materials, which is the DNA, okay, and then act as a control center that directs all activity and cell division. And then the third one, it involves in the production of RNAs, mRNA, rRNA, and tRNA, and also it is the site where ribosomes are produced, okay. Which is needed. The ribosome function is to synthesize protein within the cell. Okay, protein, enzyme, right? Hormones. So the ribosome then later involved in protein synthesis. Okay, so next we are going to look at ribosome. Before kita proceed, ada soalan nak tanya?
Madam,
Ya. Yeah.
tadi Madam tanyakan, tadi Madam katakan yang prokaryotes ni primitive cells kan?
Prokaryotes,
Jadi mereka boleh develop jadi eukaryotes tak?
Itu akan dia tengok pada teori evolution lah. Dia kalau dulu dia ada satu teori ni, kita namakan dia sebagai endosymbiotic teori, macam tu lah. Dia asalnya bakteria, okay, bakteria yang berbentuk, kalau tengok kan dalam sel eukaryotes, ni pen saya hilang. Okay, dalam sel kita ni ada apa? Ada mitokondria. Sekejap. Keep this slide. Okay. Sekejap. Dalam sel kita ada nucleus, and then ada mitokondria kan. So kalau tengok mitokondria ni, contohnya bentuk dia macam bakteria. Okay. So daripada teori endosymbiotic teori tu, dulu dia kata bakteria yang bentuk macam ni, dia merge dengan eukaryotic cell. Okay, they merge together, okay, and then they form eukaryotic cell. So itu endosymbiotic teori lah. So tak ingat lagi. Saya jumpa lah dalam ni. Oops, sorry. Tak perlu, nanti Google lah cari, ada endosymbiotic theory. So asalnya dia adalah eukaryotic cell masuk dalam dia adalah prokaryotic cells masuk dalam eukaryotic cells. So dia itulah terjadinya eukaryotic cell. Itu teori lah. Okay. Itu evolution lah kan. So siapa yang percaya evolution percayalah. Macam tu. Apa itu dah tengok boleh search. Macam tu je. Tapi tak boleh lah. Apa soalan awak tadi? Kalau prokaryotik boleh jadi eukaryotik? Tak adalah macam tu. Kalau tak sekarang ni bakteria jadilah binatang ke macam tu kan. Faham ke?
Faham.
Ada soalan lain? Ada soalan? Tak ada soalan, kita break sekejap 2 minit. Kalau nak pergi toilet ke, nak pergi ambil air ke, boleh lah. Sebentar saya cari kalau jumpa tak endosymbiotic theory. Saya nak minta tolong kelas rep S61 dengan S62. Saya ada bagi gambar kat dalam grup kelas rep. Dia terangkan pasal endosymbiotic theory. Bukan endosymbiotic tapi endosymbiotic theory. Bagi kat punya member sekarang, cuba tengok pasal endosymbiotic theory tu. Okay. So dalam teori tu dia kata sekarang ni dalam sel eukaryotes kita ada mitokondria dengan kloroplas kan. So asalnya mitokondria dengan kloroplas ni asalnya dulu adalah prokaryot. Okay. So dua prokaryot ni dia masuk ke dalam host cell. Okay, host cell which is eukaryot pada masa dulu tu lah. Okay. So bila dua bakteria ni yang berbentuk macam mitokondria dengan kloroplas ni masuk dalam host cell, lepas tu dia akan dia dah suka dah duduk dalam tu, so dia tak keluar-keluar, dia jadilah mitokondria dan kloroplas dalam sel yang ada sekarang ni. Macam tu. Itu adalah teori sahaja, okay? Endosymbiotic teori. Okay?
So maksudnya mereka merge sama-sama and become parts of the cell ataupun organelles of the cell.
Sekarang ni jadi organelle lah, organelle dalam sel.
Okay.
Okay. Itu just a theory, alright? Okay, so we are going to continue with the next organelles, okay, which is the ribosome, okay, the ribosome. So what are ribosome? Ribosomes are organelles that is composed of large and small subunit. So as you know, large and small subunit are produced in the nucleus of the nucleus, okay, which combine together with ribosomal RNA or rRNA, alright, and then in order to produce the functional ribosome, so the large and small subunit ni dia akan merge together in the cytoplasm, and then they become the ribosome. Okay, so the ribosome is the organelle involved in protein synthesis, right? So cells that are active in protein synthesis will have a large number of ribosome and a prominent nucleoli. Prominent nucleoli tu maksudnya nucleus dia tu macam tersembul keluar. They have a large nucleus, right? Okay. So example, you have pancreas. So as you know, pancreas, the function of the pancreas is to produce insulin, glucagon. So insulin and glucagon are hormones that are made up of proteins. And then another function of pancreas is to produce digestive enzyme such as amylase. So those digestive enzymes are also proteins. Okay, so pancreas is a good example of organs that have cells with large number of ribosome. Okay, because they produce enzyme and also hormones. Okay, so ribosome has two types. Okay, two types of ribosome. The first one is free ribosome, and then you have bound ribosome. So free ribosome are suspended in the cytosol. They are free in the cytoplasm. Okay, and then they are not bound to any structure, especially they are not bound to the rough ER. And then the function of the free ribosome is to produce protein to be used within the cell only. Itu keyword dia. Okay, so produce protein to function within cell only. Okay, so for example, enzyme that is used, okay, within the cell, okay, or to produce structural protein, okay, within the cell for cell growth, for example. Okay, so the next type is bound ribosome. So bound ribosome are attached to the outside of the endoplasmic reticulum or the nuclear envelope. Okay, so you have here the nucleus, okay, the inner nucleus, the inner membrane, and then the outer membrane. So the outer membrane is connected to the rough ER, for example. Rough ER, it is made up of flattened sac. Okay, so here you have the lumen of ER. Okay, so lumen of ER, kawasan dalam space, okay, within the ER, and then the ribosome are found attached, okay, to the outside of the of the endoplasmic reticulum. Outside to means that you have to draw the ribosome facing the sorry. Hilang lah pen saya ni. Ini cytoplasm. Cytoplasm, okay. You have to draw the ribosome facing the cytoplasm. Kalau awak draw ribosome ni inside di sini, awak punya lukisan jadi salah, okay. So the ribosome has to face the cytoplasm, facing outside, okay, facing the cytoplasm, not inside the lumen, alright, facing outside. So can be found here also, okay, but still in the cytoplasmic region, okay. And then the function of the ribosome, the bound ribosome, is to produce protein to be inserted into membrane or exported out of the cell, okay. So cells that specialize in protein secretion have a high number of bound ribosomes, such as pancreatic enzyme, okay. So bound ribosome will only produce protein to be used outside of the cell, or to produce enzyme that is confined within a vacuole. For example, hydrolytic enzyme in lysosome. Okay, hydrolytic enzyme in lysosome are produced by bound ribosome. Okay, enzyme produced by the pancreas are also produced by bound ribosome because it is secreted out of the cell. Okay, so you have to know the function. Okay, the difference in the function of free and bound ribosome. Free ribosome to produce protein within to be used within cell, bound ribosome to produce protein to be secreted out of the cell, or to produce enzyme of lysosome, for example. Okay, so itu perbezaan dia. Ataupun produce protein to be inserted into membrane. Protein to be inserted into membrane, we call it as glycoprotein. Okay, dalam awak punya, ini awak punya sel kan, dalam awak punya plasma membrane ni, ini adalah plasma membrane. Dalam plasma membrane awak ni akan ada satu sejenis protein, okay, that is embedded in the plasma membrane, and they have carbohydrate attached to it. So we call it as glycoprotein. Glycoprotein. This glycoprotein, okay, is produced by the ribosome, which is attached to the rough ER, and this glycoprotein will have their own specific function. One of the specific function of the glycoprotein, for example, it serves as identification tag for the cell. Okay, itu nanti kita tengok what is identification tag of cell. Itu chapter 3 lah nanti kan. So glycoprotein has their own specific function, as for example identification tag, okay, ataupun as antigen lah, macam tu, okay. Okay, so this is to show you the structure of ribosome, or the image of ribosome that is seen under electron microscope. Okay, so these are free ribosome. Okay, you find a free ribosome scattered in the cytoplasm, and then here you have the okay, this is the rough ER. Okay, rough ER, where you have bound ribosome attached to the membrane of the rough ER facing the cytoplasmic site, bukan dalam lumen. Okay, facing the cytoplasmic site. So this is the lumen of rough ER. Kita panggil dia sebagai cysternal space. Okay, cysternal space. Okay, so next is the endomembrane system. Okay, so the endomembrane system, what are endomembrane system? The endomembrane system is composed of several components. It has 6 components. the nuclear envelope, the endoplasmic reticulum. You have two types of endoplasmic reticulum, the smooth ER and the rough ER. You have the Golgi apparatus, you have the lysosome, the vesicle and the vacuole, and also the plasma membrane. Okay, so vacuole it also includes central vacuole. Okay, in the plant cell. Okay. So the function of the endomembrane system is to regulate protein traffic and also perform metabolic function in the eukaryotic cell. Okay, metabolic function, it means to synthesize products, for example. Okay, and then so this component, all these 6 component, okay, are either continuous or connected via transfer by vesicles. So it means that the membrane of all these component membrane, the nuclear membrane, the membrane of the rough ER, the membrane of the smooth ER, the membrane of the Golgi apparatus, the membrane of the lysosome, okay, the membrane of the vesicle, and also the plasma membrane itself, the membranes are connected to one another, okay, either through direct physical continuous connection or through the transfer of vesicle. So kalau tengok kat diagram ni kan. So as you can see from the diagram here, the outer membrane of the nucleus is directly connected to the membrane of the rough ER. The membrane of the rough ER, as you can see here, is directly connected to the membrane of the smooth ER here. Okay, and from the rough ER, you have the formation of transport vesicle. So the membrane of the transport vesicle was the membrane of the rough ER. Now, when you produce the transport vesicle here, which contain proteins, okay, which is produced by the bound ribosome, then fuse with the membrane of the Golgi apparatus. So now the membrane of the transport vesicle becomes the membrane of the Golgi apparatus. So the function of the Golgi apparatus is to modify protein, okay, modify the protein that is made by the rough ER, okay, ribosome which is attached to the rough ER. So the Golgi, okay, will modify the protein, and then it then package the protein into vesicle again. So this time it becomes secretory vesicle. Okay, so this one is a secretory vesicle. So the membrane of the secretory vesicle was made by the membrane of the Golgi apparatus. Okay, so now this membrane will fuse with the membrane of the plasma membrane to secrete out the protein. Okay, for example, to secrete out hormones. Okay, for example, or secrete out enzyme, for example. So as you can see, the membranes of this structure are connected directly or through the transfer of vesicle. So here, okay, daripada Golgi dia akan jadi, for example, lysosome. So the lysosome also has membrane that originates basically from the membrane of the rough ER. Okay, so basically the rough ER, we refer the rough ER as the membrane factory sebab semua membrane ni asal dia daripada membrane of the rough ER. Okay. Okay, so the properties of the endomembrane system. So it carries out a variety of tasks, okay, in cell, including 1, protein synthesis and transport protein, either into membrane and other organelles. Okay, so the function of the endomembrane system, if you can see in this diagram, the rough ER, it has bound ribosome, and the function of the bound ribosome is to synthesize protein. Okay, so the protein is now within the lumen of the rough ER. Okay, so the protein is packaged into vesicle. Okay, so that's why this it involves in protein synthesis and transport the protein into membrane or into other organelles. Maksudnya dia akan masuk ke dalam Golgi apparatus, dia masuk ke pada dalam lysosome, contohnya. Okay, so itu masuk dia dengan into membranes or other organelles, right? And then or out of the cell. Ini saya tambah dalam nota awak tak ada, or out of the cell. So the protein that was produced by the rough ER can be secreted out from the cell to be used out of the cell. Okay, to function out of the cell. Macam contoh tadi lah, insulin kan, insulin it function out of the cell. Digestive enzyme function out of the cell of the pancreas. Okay, and then the next function of the endomembrane system it involves in metabolism and movement of lipids. This one, it involves the function, basically the function of smooth ER. Okay, smooth ER involves in the synthesis of lipids, and then the detoxification of poisons. Okay, and then the last one, membranes of this system are related either through direct physical continuity. Macam tadi lah, tengok the outer membrane of the nucleus is directly connected with the membrane of the rough ER. Rough ER is directly connected with the membrane of the smooth ER, or by the transfer of vesicle. So daripada rough ER, dia akan hasilkan transport vesicle, fuse with the membrane of the Golgi apparatus, produce secretory vesicle, fuse with the plasma membrane. That is what it meant by transfer of vesicle. Okay, so that is basically the rough idea of the function of the endomembrane system. Okay, so we are going to look at the first component, which is the endoplasmic reticulum. So the endoplasmic reticulum, it is an extensive network of membranes. Okay, extensive maksudnya dia ada banyak membrane. Okay, dia terdiri daripada banyak membrane sebab struktur endoplasmic reticulum ni dia memerlukan banyak membrane lah. Okay, so account for more than half of the total membrane in many eukaryotic cells, consists of a network of membranous tubule and flattened sac called cisternae. So membranous tubule is referring to the smooth ER. It is the structure of smooth ER. But as for flattened sac, it is referring to the structure of rough ER. Okay. So this structure, membranous tubule or flattened sac, we call it as the cisternae. Okay, cisternae. Make sure you know how to spell the spelling correctly, cisternae. Okay, so inside the cisternae you have space. Okay, we call it as cisternal space, which is the internal lumen or space of the ER, which is separated from the cytoplasm by the ER membrane. So the ER membrane is a single layer membrane. So it is continuous with the outer membrane of the nuclear envelope, okay, and then separates the nuclear lumen or the cisternal space from the cytoplasm. Okay. So this is the cisternal space, okay, yang ruang dah ni, cisternal space, cisternal space. So the protein are synthesized, okay, by the bound ribosome, and they are synthesized within the cisternal space. Okay, so there are two distinct regions of the ER. Smooth ER, why do we call it as smooth ER? Because they do not have any bound ribosome. Lack, okay, means it does not have at all. Okay, bukan kurang tapi lack, does not have any at all ribosome attached to it. Okay, and then the next one is bound ribosome. They have bound ribosome lah. Okay, rough ER have bound ribosome attached to the outer surface of the membrane. Okay, so this is the region of the smooth ER. So this is the region of the rough ER. Okay, so rough ER has bound ribosome attached to it. So smooth ER, they lack ribosome. Lack bukan maksud kurang, lack maksudnya tak ada. Okay, does not have any at all ribosome attached to the smooth ER. So it is a system of interconnected tubule. So ingat ya, the tubules is also referred to as cisternae. Okay, so it is continuous from the rough ER. So the function of the smooth ER is totally different from the rough ER. Okay, one is to synthesize and transport lipids. So these lipids include oils, membrane phospholipid, and also steroids, hormones. So for example, cells of testes and also ovaries will have high number of smooth ER. Why? Because these two organs involved in the synthesis of sex hormones, okay, which is made up of steroids. Okay, so that's why they have large number of smooth ER. So ini contoh lah. And then another function of smooth ER is that it involves in metabolism of carbohydrates. Okay, for example, the liver. Okay, liver cells. Function of liver cells is to store glycogen. Okay, is to store glycogen. So that's why the liver will have a large number of smooth ER. Okay, and then another function of a smooth ER is to detoxify drugs and also poison. So this is also done by the liver. And then another function of the smooth ER is to store calcium. Okay, so calcium in the muscle cell. Okay, so muscle cell will have to have calcium, okay, in order for the muscle cell to be able to contract. Okay, so in order to allow the muscle cell to contract, so they have to have calcium. Okay, so the next endoplasmic reticulum is the rough endoplasmic reticulum. So consists of cisternae, which is an interconnected system of membrane-bound flattened sac. Okay, so ingat ya, kalau rough ER, flattened sac. Kalau smooth ER, interconnected tubule. Kalau tengok structure dia ya, so this is tubule, untuk macam tube. Tapi kalau rough ER, flattened sac. Okay. Membrane-bound flattened sac. It is continuous with the outer membrane of the nucleus. They have many ribosomes attached to the outer surface of the membrane. So it is abundant in cells which are rapidly growing. Okay. Or in secretory cells. Okay, secretory cells. Example of secretory cells is pancreatic cells. Secretory cells, it means that cells that produce protein to be secreted out of the cell. Okay, pancreas produce enzyme, secrete the enzyme out of the cell. Pancreas produce hormones that is secreted out of the cell. So that's why we call it as secretory cell. Okay, and then cells of the salivary glands. Salivary glands secrete enzyme. Enzyme apa? Enzyme amylase. Okay, so this amylase is secreted into your oral cavity, okay, to digest carbohydrate that you eat, okay, rice for example, bread for example, okay. So that is the function of the amylase secreted by the salivary gland, okay, which is produced by the rough ER. Ini saya punya explanation untuk rough ER ni panjang sikit ya, awak punya nota tu dia macam summarizekan sikit. Alright, so it says here, bound ribosome synthesize polypeptide chain. So this is the cisternae of the rough ER, and then outside you have the ribosome, okay, the bound ribosome. As you can see from the diagram here, the bound ribosome will synthesize protein into the lumen of the ER, the cisternal space. Yang purple color here is the cisternal space. The blue thread here is the protein synthesized by the ribosome, which is indicated by the green structure here. Okay, the ribosome. So ribosome synthesize the protein into the lumen of the ER. Yang merah ni adalah mRNA. In order for the ribosome to synthesize the protein, it has to be from the instruction of the mRNA. Okay. So as the new protein enters into the lumen, okay, it is folded into a specific shape, okay, native shape, it means that into specific shape, okay, and then therefore the cells secrete proteins that are produced by ribosome attached to the rough ER, okay. Rough ER is basically to produce protein to be secreted out of the cell because they are packaged, okay, into transport vesicle that will later fuse with the plasma membrane, okay. And then an example is pancreatic cell. So pancreatic cell is to produce insulin, which is a type of hormone made up of protein, and then the hormone is secreted into the blood stream, okay, to regulate blood glucose level, okay. So nampak so insulin it function out of the pancreatic cell, okay, function out because this protein will be secreted out, okay, from the pancreatic cell, secreted out, function out of the cell, okay, which is to regulate your blood glucose level in this example, okay. So most secretory proteins are glycoproteins, okay. What are glycoproteins? Proteins that have carbohydrates covalently bonded to the protein. So the carbohydrates are attached to the protein in the ER lumen. So as the as the protein are synthesized, they are folded into specific shape, and you have carbohydrate attached to the protein to form the glycoprotein. So, so it says here the secretory protein produced are moved to a specialized region of the ER, which is, we call it as the transitional ER. If you look at your diagram of the rough ER, they have a special region which is called as the transitional ER, okay, the transitional ER. So it means that in this region, the transitional region is where the transport vesicles are produced, okay. So these transport vesicles will contain protein, okay, protein produced by the bound ribosome or the glycoprotein. Kalau awak tengok kat sini kan, bila awak enlarge kan dia, macam mana nak imagine glycoprotein tu? Okay, so this is the membrane of transport vesicle. Okay, this is the membrane of transport vesicle. So this is the protein. Okay, the protein of which the protein is bonded to oligosaccharides. So now it becomes glycoprotein. So this is a glycoprotein. So as you can see from this diagram, the green structure here, which is the carbohydrate region of the glycoprotein, is facing inside of the vesicle, of the transport vesicle. Tapi nanti kan dia akan pergi kepada Golgi apparatus to be modified. Apparatus to be modified, and then from the Golgi apparatus dia akan package balik semula dalam bentuk secretory vesicle. So dia akan modify the protein, the glycoprotein. Okay, modify the structure so that it becomes more functional. For example, so as you can see in this secretory vesicle, namakan dia secretory vesicle. So this secretory vesicle, imagine kan, ini sebab saya tak muat dah nak draw, saya draw dia kat sini lah. Kat sini ada secretory vesicle. Okay, secretory vesicle yang mana ada the glycoprotein kan. So dia akan fuse. Okay, dia akan fuse dengan membrane of the plasma membrane. Dia akan fuse dengan membrane of the plasma membrane. Dia akan fuse macam ni. Dia akan fuse. Diagram saya pun saya tak buat. So dia akan fuse. So bila fuse, tengok the glycoprotein now, okay, asalnya dia facing inside of the vesicle, now it will face the extra fluid region. So ini adalah region yang ada extra fluid region, extracellular fluid region. Okay, so now the glycoprotein will serve as for example identification tag for the cell. So identification tag tu maksudnya dia akan membezakan, sel ni adalah berbeza dengan sel yang ini. Sel ni adalah berbeza dengan sel yang ini. Macam contohnya dia become antigen lah. Macam contohnya kalau red blood cells, it becomes the antigen. Okay, for example. Okay. So initially the glycoprotein was inside of the cell. It was inside of the secretory vesicle. But when the secretory vesicle fuses with the plasma membrane, the glycoprotein will face the extracellular fluid. Okay, it is embedded in the plasma membrane but facing the extracellular fluid side. Okay. So next, the vesicle containing the secretory protein will bud off from the ER membrane. Ini maksudnya bud off, right? Dia terkeluar daripada membrane of the vesicle and travel to the Golgi apparatus for further processing, okay, for modification, okay.
Madam nak tanya?
Ya.
Maksudnya yang glikoprotein ni dia keluar tak daripada sel?
Tak, dia jadi part of the membrane.
Oh, okay.
Okay. Dekat awak punya plasma membrane akan ada protein, okay? And one of the function of the protein, dia akan jadi contohnya glycoprotein, ataupun dekat awak punya plasma membrane akan ada protein yang bertindak sebagai enzim, ataupun dia akan jadi protein yang bertindak sebagai transport protein. All these protein that are embedded in the plasma membrane was originally made from the RAF-ER. Okay, sama juga kalau contohnya membrane of the itu je lah. Okay, so your plasma membrane ada banyak jenis protein dan protein-protein ni ada banyak fungsi. So in order for your plasma membrane to have so many different types of protein, so the protein was originally made by the RAF-ER. Itu je dia nak cakap. Okay. Itulah chapter 3 lah, kita akan tengok nanti. Okay, so the RAF-ER also acts as membrane factory. Dalam satu slide awak tu kan, yang poin bawah sekali dia cakap RAF-ER also acts as membrane factory, okay, factory which is to manufacture more membrane for the cell. Okay, so it is done by adding membrane proteins and also phospholipids to its own membrane in making the secretory proteins. Okay, so therefore the ER membrane expand and is transferred in the form of transport vesicle to other component of the endomembrane system. So all the membranes of the endomembrane system was originally from the membrane of the RAF-ER. Tadi kalau tengok kan macam gambar tadi tu. Okay, so this basically it shows you the membrane of the endomembrane system, the component of the endomembrane system. So the membrane of the nucleus, the membrane of the endomembrane itself, or the smooth ER, or the membrane of the transport vesicle, or the membrane of Golgi apparatus, or the membrane of the the lysosome, or the membrane of the plasma membrane was originally made by the RAF-ER. So RAF-ER, that's why dia adalah bertindak sebagai membrane factory. Okay, it is the membrane factory for the cell. Okay, so the ER membrane will expand, right? And then dia tak terus expand kan, sebab dia akan transfer the membrane to transport vesicle to fuse with membrane of Golgi or membrane of the plasma membrane, or becomes the membrane of lysosome, for example. Okay. So ni satu lagi gambar yang saya tambah, sama je, alright. So this is the ER, okay, you have the RAF-ER where you have the ribosome attached facing the cytoplasmic side, and then the ribosome will synthesize protein. So nampak protein, the green structure here will fold into specific shape, okay. And if it's become glycoprotein, you have carbohydrate which will attach to the protein, and then the protein will go to the transitional region of the ER and package into transport vesicle, and then the transport vesicle will bud off from the transitional region, and then they can fuse with the membrane of the Golgi apparatus. If you see from this diagram, the Golgi apparatus, it has two major region, okay, which is this region we call it as the cyst phase. Cyst phase is the region of the Golgi apparatus facing the rough ER, okay. Cyst phase. So cyst phase will receive the transport vesicle. So the transport vesicle will fuse with the membrane of the Golgi apparatus, modify the protein, okay, modify until the protein reaches the trans phase of the Golgi apparatus. Trans phase of the Golgi apparatus is the region of the Golgi apparatus that faces the plasma membrane. Okay, so from this region, okay, after the protein has been modified, fold into specific shape to become functional protein, for example, enzyme. Okay, so the enzyme, okay, will be packaged into this vesicle. We now call it as the secretory vesicle. Kalau kat sini kita panggil dia sebagai transport vesicle. Kalau kat sini sebab dia akan fuse dengan plasma membrane, we call this as secretory vesicle. So the vesicle will fuse with the membrane of the plasma membrane and secrete out the product. So that's why tadi kita tengok one of the function of bound ribosome is to produce protein to be secreted out and function out of the cell. Okay. Okay. So next you have the Golgi apparatus. So basically the function of the Golgi apparatus, it works as shipping and receiving center. Okay, basically receive dulu lah, it receives the protein from the rough ER, okay, and then modify the protein and then package the protein into the secretory vesicle and ship the protein to be secreted out by the vesicle fusing with the plasma membrane. Okay, so that's why dia bertindak sebagai receiving, modifying, packaging and shipping proteins. Okay, and then consists of flattened membranous sac called the cyste. Ni sama juga macam rough ER or smooth ER. So the flattened sac is also called as the cistern. And then each Golgi sac has three areas. Okay, so the cyst phase is the region where it faces the rough ER. And then transphase is the region that faces the plasma membrane. And then you have the middle region, the middle region of the rough ER. So product from the rough ER, example glycoproteins, are modified during the transition, during the transit from cis to trans. So the protein that enters into the Golgi apparatus will be modified. Okay, modified maksudnya dia akan fold into a specific shape, or it adds more carbohydrate to it. Alright, so the protein becomes more functional. Okay, and then once you have a functional protein, then the protein will be packaged into vesicle, which is this time we call it as secretory vesicle. Okay, so the glycoprotein are packaged in transport vesicle in the trans phase. Okay, awak boleh panggil dia sebagai transport vesicle juga, tapi bila dia nak secrete product, okay, then we call it as secretory vesicle. Okay, so in this diagram, okay, so transport vesicle, okay, this one, transport vesicle or secretory vesicle, okay, which departs from the trans phase, contains secreted products, okay, inside, and then it fuses with the plasma membrane. Okay, so now the membrane of the vesicle becomes the membrane of the plasma membrane. So the function of the Golgi apparatus is to modify products of the ER. Products of the ER was the protein or the glycoprotein produced, and then manufacture certain macromolecules and also sort and packages materials into transport vesicle. Okay, transport vesicle ataupun secretory vesicle. So sama je, this explanation goes to this diagram. Okay, the same explanation. So you have here what? The transport vesicles that is produced by the rough ER. Okay, vesicles move from rough ER to Golgi. Okay, and then vesicle merge with the membrane of the Golgi and becomes now the membrane of the Golgi apparatus. Okay, or the Golgi cistern. And then the cisternal maturation. So Golgi cistern move in the cis to trans direction, cis to trans. Okay, daripada region facing the ER, and then it reaches the region where it faces the plasma membrane. So vesicle form and leave Golgi carrying specific products to other location or to the plasma membrane for secretion. Okay, so other location is referring to, for example, the Golgi apparatus will produce hydrolytic enzyme. Okay, so this hydrolytic enzyme is packaged inside lysosome. Maksudnya untuk hasilkan lysosome adalah daripada Golgi lah. Okay, so for example, this vesicle will become lysosome later. Okay, so the lysosome will function elsewhere in the cell. Okay, and then vesicle transport to some transport some protein backward to less mature Golgi cisternae where they will function. And then or vesicle also transport certain proteins back to their site of function. Okay, so macam tadi lah, ada protein bila dia embedded dalam dalam membrane of this vesicle, dia boleh bertindak sebagai ini kan. Sekejap. Okay, kat sini akan ada protein, for example. The protein that is embedded in the membrane of this vesicle can become enzyme. Okay, it can become enzyme. So when this enzyme or the protein that was embedded in the membrane of vesicle can now become the enzyme that function in the membrane of the ER. Okay, contohnya. So same explanation nampak kat sini kan. So number one, where is number one? Polypeptide synthesized on the ribosome. Okay, so polypeptide yang ni lah kan, polypeptide synthesized on the ribosome. So you produce protein by the ribosome attached. So protein assembled and carbohydrate component added in the lumen of the ER like this one. Okay, and then transport vesicle move glycoprotein to Golgi from cis to transphase. Okay, so glycoprotein are further modified in the transphase. Glycoprotein package in the transport vesicle. So this is it becomes the transport vesicle. So glycoprotein are transported to the plasma membrane and content released from the cell. In this case, it becomes glycoprotein lah, kan, which is embedded in the plasma membrane. Ada yang embedded, ada yang secreted out. Okay. So the next organelle okay, so the next organelle is the lysosome. Okay, so what is a lysosome? Lysosome is a membranous sac of hydrolytic enzyme that can digest macromolecules. Okay, so inside the lysosome you have hydrolytic enzyme. Okay, hydrolytic enzyme. So lysosomal enzyme can hydrolyze protein, fats, polysaccharides, and also nucleic acid, and they will work best in acidic environment, which is pH 5. If lysosome breaks, the released enzyme are not effective as cytosol has a natural pH. Okay, so but however, if you have excessive leakage of this hydrolytic enzyme from so many lysosome, okay, so this can lead to auto digestion. Okay, so the next one, okay, lysosome carry out intracellular digestion. For example, phagocytosis. What is phagocytosis? A process by engulfing food particles. Food vacuole form and fuse with lysosome, and the enzyme digest food. Contoh phagocytosis yang ni. Okay, so outside of the cell, so this is a cell. Outside cell you have food. Okay, so this cell will start to engulf by forming food vacuole. Okay, engulf, form food vacuole. So this food vacuole will fuse with the lysosome. Okay, the lysosome contains digestive enzyme, and this enzyme will digest the food. And then after digestion, the food particle will be absorbed into the cell to be used. Okay. Then the next one is autophagy. What is autophagy? It is to recycle cells' own organic materials. So, for example, within your cell, you have worn out organelles, okay, which is organelles that is not functional anymore. So, through autophagy, okay, so this organelle will be surrounded by membranes, and the lysosome will bind with the vesicle containing the worn out organelles, and then we'll digest that worn out organelle into smaller molecules, and then the molecule will be reused, okay, inside cell to be reassembled and used in another form. Okay, so that is autophagy. Recycle cells own organic materials. Damaged organelles become surrounded by membrane macam ni, kan? So this is worn out organelle. So it is surrounded by vesicle. Okay, so the vesicle then fuse with the lysosome. Okay, and then the hydrolytic enzyme within the lysosome will digest the worn out organelle, for example. Okay. So ingat phagocytosis is to digest food within food vacuole. Autophagy is to recycle cells on organic materials. So for vacuole, you have three types of vacuole. You have food vacuole, contractile vacuoles, and also central vacuole. Central vacuole kita dah cover tadi, which is contained within plant cells, which is, for example, to store water and other organic compounds. And then by having or storing water, it can cause the cell to be in a turgid condition, which is the normal condition for plant cell. And then the membrane of the central vacuole, we call it as the tonoplast. And then the next one is the contractile vacuole. So contractile vacuole are found in many freshwater protists. Okay, so the function of the contractile vacuole is to pump excess water out of the cell, and food vacuole are performed by phagocytosis. Phagocytosis is engulfing food, okay, and package the food into food vacuole, right? So this is the structure of a contractile vacuole of a paramecium. So paramecium is an example of protists. Okay, so they also contain food vacuole. So this is central vacuole, which contain large amount of cell sap or water that causes the cell to be turgid. So this is just to summarize, okay, the relationship between the component of the endomembrane system. So you have to remember the endomembrane system consists of the plasma membrane, the nuclear membrane, okay, which is directly connected to the membrane of the rough ER, which is started by the ribosome to produce protein, and then the protein will be packaged into transport vesicle in the transitional ER, okay. And then this transport vesicle will fuse with the membrane of the Golgi apparatus at the cyst phase, okay, of the Golgi, and then the protein will be modified, okay, as it travels through the Golgi until it reaches the trans phase of the Golgi. So the protein now will be packaged into a vesicle. So if the vesicle becomes another organelle, for example, lysosome, it contains hydrolytic enzyme, for example. This one, it becomes secretory vesicles, okay. So the secretory vesicle will fuse with the plasma membrane to secrete out the product, okay, or to cause glycoprotein to be embedded in the plasma membrane facing the extracellular fluid side, okay. So that is the end of the endomembrane system. Ada soalan? Tak ada soalan ke? Semua faham
Faham tadi?
Saya nak yes. Sekejap. Move to. Ini ada attendance, tolong isi. Ni soal tanya apa beza cis Golgi dengan trans Golgi. Cis Golgi dia face, kalau tengok gambar ni, the cis part of the Golgi it faces the rough ER. Okay, so dia adalah region pada Golgi yang akan accept ataupun terima transport vesicle yang datangnya daripada rough ER. Kalau trans phase of Golgi, dia akan phase ataupun lebih dekat dengan plasma membrane. Sebab apa? Sebab daripada trans phase of Golgi, dia akan hasilkan secretory vesicle contohnya. Dalam secretory vesicle ada protein yang akan dikeluarkan daripada sel to be secreted out. Okay? So this is trans phase. Trans phase is much more nearer to the plasma membrane because it will produce secretory vesicle containing protein to be secreted out of the cell. Itu beza antara cis and trans phase. Phase tu bahagian, region, okay? Region pada Golgi. Boleh Anis? Okay. As 62 ada tutorial kan dengan Sir Helmi, so make sure you sign the attendance dan awak bolehlah leave the meeting dan yang lain pun sama juga. Okay, terima kasih. Kalau tak ada soalan, kalau ada soalan boleh keep on tanya sementara tunggu yang lain live lah.
Thank you, Madam.
Okay.
Thank you, Madam.
Okay.